So for the DNA replication, right? So what we're basically going to be doing is chilling in interface for a while, right? And then we are going to be preparing for some amount of DNA replication, right? So we're gonna have our initiator proteins binding to the origin, right? And then causing our helicase to come in to break those bonds. And we're gonna have single strand DNA binding proteins that will bind to make sure that the hydrogen bonds are not replaced, all right? For primer binding now, we're gonna have the addition of primers, right? So after we have all of that now, at the origin of replication, helicase will start to now break the bonds, right? While the single strand DNA binding proteins are gonna maintain the open fork, right? The open area. Primase, right, an RNA polymerase enzyme would synthesize a short RNA primer complementary to the DNA template strand, right? This primer provides a free 3' end to which DNA polymerase can add nucleotides. So what we're doing, we're starting off the polymerase using primase, adding that 3' primer, all right? That free 3' end primer for no polymerase to now pick up on, right? And here we now have an elongation where the DNA polymerase now, right, that got onto the primer itself, right? That DNA polymerase now will catalyze the addition of the deoxyribonucleotides to the 3' end of the growing DNA strand. Alright, we're just looking at it in a little bit. Alright, so the 3' end. So the direction of synthesis from 3' going upwards there. Right? So the leading strand synthesis DNA polymerase continuously adds nucleotides in the 5' to 3' direction, moving towards the replication fork. We're gonna be looking at another diagram, right? And the lagging strand now, the DNA polymerase synthesis synthesized rather short fragments of DNA called Okazaki fragments, also in the 5' to 3' direction away from the replication fork. So what's happening here now? We have helicase there continuing to go through, right? We have our primase there and our RNA primer. So what's happening here is that when the primers are placed, right, the DNA polymerase, right, would actually go through to create Okazaki fragments, right? I'm not sure if we're going to be speak. Yeah, we're gonna be speaking about that. So the creation of the Okazaki fragments, right? What's happening here is that DNA polymerase 3 would actually be the one creating the leading strand. So completely fine. It's just a smooth movement along the leading strand. While with the Okazaki fragments, no, the creation of the Okazaki fragments there would be by DNA polymerase 1, right? We should actually build, right, in the opposite direction away from the leading strand, right? So while the leading strand, right, with the leading strand, we just build directly in the just smoothly in one direction, right? What DNA polymerase has to do, right, is build a fragment, then drop off, then build another fragment, then drop off, then build another fragment, then drop off. So it's actually slower than that of the leading strand down here, right? So the leading strand is actually going to create smooth synthesis, smooth polymerization, right? Which is much quicker than what we have for the lagging strand, which we have Okazaki fragments on, right? And we actually are going to have some areas where there are no actual bonds, right? And for that, no, we're going to have to look at the other polymerases to fill those areas and also to have DNA ligase to actually bind the proteins together. Well, why am I calling them proteins? The nucleotides together. All right. So if we look at this now, how do we process these Okazaki fragments? Because they're in tiny fragments, they're not like the leading strand that creates generally one main strand, right? So the DNA polymerase synthesizes the Okazaki fragments discontinuously, resulting in RNA primers interspersed between the DNA fragments, right? So in between the DNA fragments, you're gonna have RNA primers, right? The polymerase 1 removes the RNA primer and replaces them with DNA nucleotides, right? So in the case down here, let's look at this place down here, right? We're gonna have an Okizaki fragment here. The red parts would be a primer, right? So let's say that the red part here is a primer, and then we have the brown part, which is the bases, and then we have a primer here, and then we have bases here, right? What RNA polymerase 1 would do is remove that primer, the RNA bases, replace them with DNA bases, and then DNA ligase will come in now and seal those joints between the Okazaki fragments, creating a continuous strand, right? So that is the treatment of the Okazaki fragments. Mm-hmm. Okay. For termination, however, no. Replication continues bidirectionally, right? In two directions, we have the leading direction, leading strand, and then the lagging strand, right? Until the DNA molecule is copied. In eukaryotes, termination occurs when the replication fork from neighboring origins of the replication meets. And in prokaryotes, replication terminates when the replication fork meets the termination site. All right. So generally here, so we're looking at all the stuff what's happening here, right? So we have helicase. Okay, we have the first the initiation proteins that create the space for helicase to come in and unwind, right? Helicase will follow where topoisomerase is. What topoisomerase will do is straighten the chain so helicase can break it, right? Then we're going to have the creation of our well, two different strands. DNA polymerase 3 will create the leading strand, while on our lagging strand, we're going to have RNA primers, right, created by primase, right, which will then be followed by DNA polymerase, which will create Okazaki fragments and leave leave different divots and nicks in the strand itself, right? In order to fix those issues now, DNA polymerase 1 would replace the RNA substituents with DNA substituents, and then DNA ligase will now bind them together. And that's how we treat our lagging strand, or that's how the lagging strand becomes one continuous strand, right? And that's what's happening there, right? So having a look at this now, gonna have that. We're gonna have a leading strand, we're gonna have a lagging strand, right? Just showing how it looks with this interpretation here. Okay. Okay. So let's have a look at this interpretation now. Right, so this is a better interpretation of what's happening here. So in some cases, you're going to see DNA polymerase 3 be called DNA polymerase sigma, and then polymerase 1 would be called alpha, but don't have to really focus on that. Just polymerase 1, polymerase 3, right? You know the different stuff. So I want you guys to mention the use of topoisomerase, helicase, the two DNA polymerases, polymerase 1 and polymerase 2, DNA ligase, RNA DNA primase as well, right? Generally those enzymes that help in this reaction here to actually synthesize our new strands. Okay. No, we have to speak about the semi-conservative replication now. There's a semi-conservative nature to the replication of DNA, and it basically speaks to the fact that where each newly synthesized DNA molecule consists of one parent strand and one newly synthesized strand. So this ensures faithful transmission of genetic information from one generation to the next and provides a mechanism for genetic diversity through mutation, right? So here now we have the parent strand, it's segregated and creating the two separate strands through what we just described as DNA replication. And what we're going to end up here now is going to have a semi-conservative model. Semi mean half of it is conserved, right? So one strand is conserved and one strand is new. And then if we go to a next generation now, we're going to have completely new DNA strands now, and some are conserved. All right. So generally, the semi-conservative nature of DNA, that is what's happening here, right? It's to ensure that we have the faithful um, replication to make sure that it's actually the same chain that we're looking at. Because if it's not the same chain, if it wasn't replicated properly, then it will not form a perfect helix because some base will be out of line. But if you have the complete helix being formed, that's how you generally know that everything is fine. All right. So that should be fine. And at the end of this now, what we're looking at now is the central dogma of genetics, right? But before we get to the central dogma of genetics, are there any questions or issues? Because everybody seemed like everything is fine. Through DNA replication, everything is fine. Alright? This is probably just a review you guys wanted, right? Just a review. Okay? So, the central dogma of genetics, now we know the central dogma of genetics to be the fact that DNA, right? DNA codes for mRNA, which codes for proteins. That is the central dogma of genetics. And what the central dogma of genetics is speaking about is the fact that our genotype determines our phenotype. Our DNA would determine the proteins we create. So when there's an issue in our DNA, then our proteins will change. If there's a mutation in our DNA, then our RNA will code for the mutated DNA, which would actually produce mutated proteins. Right? So that's something that's important. How does our DNA structure relate to our phenotype? Because our DNA would produce mRNA that will produce proteins that are expressions, right, of our DNA sequence itself, right? So if our DNA sequence is different, our proteins are going to look different because their RNA sequences are different, right? So that's how we really look at it. Usually humans have around 99.9 something percent of our DNA across all humans on this very planet are literally exactly the same. But that small percentage that differs changes us to be completely different individuals, except for in the case of twins. That's a little bit different, right? But in the case of general humans now, we literally have majority, right, of our DNA the same. But that tiny percentage in which we differ, it codes for different types of proteins, right? Are allowing for different variations, right? So what we're looking at really is just a central dogma of DNA and how it relates to how organisms present, right? Because of their genetic information, right? You are the way you are because your DNA sequence goes in a particular fashion. That is it. There's nothing more to it, right? So if your DNA sequence were to change, you could look differently. Your proteins may be a little bit different. You may develop genetic diseases as mutation will cause different types of changes in our sequences, which will affect how our proteins are created or what type of proteins will be created. So what I'm doing now is just really relating it back to that now, right, with the central dogma of genetics. All right. So that's really our review there on DNA synthesis or DNA replication.